Hi, everyone, and thanks for listening. This is a talk titled Phylogeography of Amansia glomerata in the Hawaiian Archipelago. I am Jimmy Fumo, and my advisor is Allison Sherwood at the University of Hawaii at Manoa. So let me start out by introducing the species Amansia glomerata. It is a red algae in the order Ceramiales, the family Rhodomelaceae, with a distribution from about Madagascar to Hawaii. There is a bunch of diversity within the species that actually has about a 5% genetic difference between samples across the archipelagos of its range. And it's distributed really broadly for a single species. In the Hawaiian archipelago, we have samples from each island of the main Hawaiian Islands, although I'll get more into that in a second. We also have samples from across the Pacific and some samples from the Indian Ocean as well. This study uses the COX-1 marker to look at the differences between individuals. So there's been a lot of work done in the Hawaiian Islands about the importance of the channels as barriers to distribution for fish and invertebrates, but little work has been done in this regard for the algae. So what we've done is collect each one of these black dots as a collection point for Amansia glomerata. You can see here on the island of Oahu, we have quite a few samples collected from all sides of the island. However, in places like the Big Island of Hawaii, we need to collect more samples. But an FST is essentially a degree of variability across a population. So in this case, for instance, let's look at this orange box on the far left of the plot on the bottom. We see a value of 0.45. So a value of 1 here would indicate that these two populations are completely separate from each other. And a value of 0 would mean they're completely intermixed. So this is a significant FST value. The orange on the right side is the p-value. And we can see from this that there's the channels are actually a significant barrier to distribution for each one of these channels, with the exception of the channel between Oahu and Kauai. We have plans in the works for trying to collect some more samples from Kauai, however. We can also use this data to look at the haplotype networks that can be created from the Hawaiian archipelago itself. And we see that there's quite a bit of variability within these islands. There are certain places where a single haplotype dominates a region, for instance, this deep red color on West Maui. And there are certain places where we have this sort of light green color up at Pearl and Hermes, Liskiansky, and Laysan in the Northwest Hawaiian Islands, where a semi-broad range is occupied by a single haplotype. This pink haplotype seems to be pretty well represented within the main archipelago as well. Another interesting note is that the type specimen for this was actually collected in 1822 from the Sandwich Islands, which is what the type specimen says, which is now the Hawaiian archipelago. And it matches this white clade from Makua Beach on the west side of Oahu. So it's likely that that sample was not only collected from Oahu, but potentially from the west side of Oahu. We are in talks right now with people from around the Pacific to send us more samples of Amansia glomerata, and we are also collecting samples from here in the Hawaiian archipelago to compare and add into this network. With that, I'd like to thank my lab and everyone else who's sending samples. Thanks for listening.